So let's take a closer look at the process of drug discovery. When we've been talking about the process of drug discovery, we specifically focused on this back end with the FDA-regulated processes of human clinical trials. Our analysis of the hepatitis C drug candidate showed us that this beginning end can also be quite laborious. So let's take a deeper look at the beginning of the process. So the beginning of the process involves identifying a biological disease that needs a treatment strategy, and then within that disease state, identifying a novel target that can be used as the anchor for developing a treatment. So how do you go about identifying a good biological target for drug development? One characteristic that the target needs to have is an involvement in the disease process and little involvement in other processes. A good example that we've already seen is the bacterial cell wall. By disrupting enzymes required for the synthesis of the bacterial cell wall, we can effectively destabilize a bacterial pathogen without causing lasting harm to the host. Of course, beneficial bacteria may also be killed in the process and need to be replenished following the illness. However, antibiotics have been quite effective in part due to their selectivity for the bacterial culprit without causing major side effects to the infected person. A good drug target also needs to primarily be distributed to the diseased tissue and not to the healthy tissue. Many cancer treatments try to target specific features on the tumor cells while limiting their impact on normal tissue. The HER2 receptor is a good example of a cellular receptor that's overexpressed in many breast cancer tumors. Thus, by making a specific antibody that recognizes this receptor and then attaching a toxic drug molecule to the antibody, you can develop a highly selective treatment strategy that brings the desired drug treatment to the tumor cells and leaves the individual's normal tissue much less affected by the toxic drug. Finally, the target needs to be accessible to the drug. This can mean the drug delivery to the correct location, the half-life of the drug in the body. For example, can it make it to the correct site in the body? And when it gets there, can it have access to the protein target to bind effectively? For example, if a drug has to pass through the blood-brain barrier to access its protein target, this may be a critical hurdle for drug development and effectiveness. So once the drug target has been identified, the next step is to come up with a drug candidate. The discovery of new chemical compounds that have biological activity are known as hits. They are often found by repurposing or testing a battery of known compounds against a new target or biological activity. These compounds are often housed in what's called a chemical library. So to screen a library, it is typical to have a high throughput screening assay that allows a researcher to quickly conduct millions of chemical, genetic, or pharmacological tests. The key labware or testing vessel of the high throughput screen is the microtiter plate. This is a small container, usually disposable and made of plastic, that features a grid of small open divots called wells. In general, microplates for high-throughput screening have either 96, 192, 384, 1536, 3456, or 6144 wells. Most of the wells contain test items depending on the nature of the experiment. These could be different chemical compounds dissolved in an aqueous solution of dimethyl sulfoxide. The wells could also contain cells or enzymes of some type that have a screening assay associated with them. And the other wells then may be empty or have pure solvent or untreated samples, and they would be used as experimental controls in the assay. So you can see that this is a carousel, and each one of these things in here is a screening microtiter plate. It's barcoded to let the researcher know which set of chemicals in this library are going to be screened for an assay. So natural product libraries are derived from the biological world often producing complex structures that are difficult to synthesize from scratch. They can come from biological organisms, where compounds are isolated from things like plants, marine organisms, sponges and corals, fungi, and bacteria. They also extend to natural product-like synthesis, where some libraries are inspired by natural scaffolds, but are synthetically produced to be more drug-like or easy to manufacture. Many modern chemical libraries are generated through advanced laboratory techniques that include combinatorial chemistry, which use systematic chemical reactions to combine building blocks or reagents into every possible combination, rapidly creating thousands to millions of distinct compounds. DNA encode libraries, or DELs, are a more recent option that provide a specialized form of synthesis where each chemical is attached to a unique DNA barcode or sequence. This allows researchers to synthesize and screen billions of compounds simultaneously in a single tube, and then trace that chemical identity by reading the DNA barcode that is attached to it. Pharmaceutical companies often maintain legacy libraries consisting of every molecule they've ever synthesized during decades of past research. They can then use these legacy libraries in new high-throughput screening assays. Commercial vendors may also sell their collections to researchers that wish to screen a library for a novel interaction with a specific biological target. Researchers can also use virtual or in silico libraries, which are computer-generated collections rather than physical samples. Computer databases, such as libraries like Zinc or ChemBL, exist as digital files that contain the structures of billions of molecules. Once a drug target is identified, in silico screening can be used to determine the optimal properties of an imagined drug molecule. This novel chemical skeleton can then be sent to an organic chemist for on-demand synthesis. In this way, researchers use virtual screening to find a promising candidate in a digital library and then pay a vendor to synthesize only that specific molecule for physical testing. Here is an example of a screening assay to identify inhibitors of a process. In this assay, a protein target is attached to the surface of a well plate. A fluorescently tagged molecule that will bind with the target is also provided and serves as a positive signal in the plate. Each well of the plate is then exposed to a different compound from a chemical library, and the plates are washed and then assayed for fluorescence. Wells that have an absence of the fluorescence indicate that the chemical molecule from the library has displaced the fluorescently targeted molecule, inhibiting the interaction. The high-throughput screening assay will then score positive hits as those that have lost fluorescence, in this case, and can then identify the compounds showing this activity. High-throughput screening is a relatively recent innovation made feasible largely through modern advances in robotics and high-speed computer technology. It still takes a highly specialized and expensive screening lab to run a high-throughput screening operation. So in many cases, a small to moderate-sized research institution will use the services of an existing high-throughput screening facility rather than setting one up for itself. And this just showing you a 96 well plate that has a positive result and would indicate that you would have a hit. So we said that discovery of new chemical compounds that have biological activity are known as hits. They're often found again by repurposing or testing a battery of known compounds against a new target or biological activity. And these compounds then came from our chemical library that we were just discussing. So once you've done your screening assay and you've identified your apparent hits, you're going to need to remove ones that show up as false positives. This can be done by an assay called PAINS, or a pan-assay interference compound. This can use computational filtering that will flag substructures that commonly elicit a false positive. Other follow-up assays can be used to confirm an initial hit and provide target-specific validation. Other assays may look for structural fragments that are similar to the structure of the initial hit and analyze these for activity as well. These help to identify the true hits within your pool and can lead to quality lead compounds that you can use for further testing. Once these quality candidates are identified, they can go into the drug optimization phase, which will be the subject of the next lecture.